We now go to protein structure. So if we are familiar with the structure of a protein, we are getting close to becoming friends with that protein. We will be able to predict their function, for example. Now, protein structure has a hierarchy. So there are levels of protein structure. And as we go along these levels, you will see why people had to create this hierarchy of protein structure. So there are four levels in this hierarchy. We have the primary structure, secondary, tertiary, and quaternary structure. So I will just mention a few details for each level. Let's go to the primary structure. When we say primary structure of a protein, we are talking about its amino acid sequence. So, for example, here we are shown insulin from a bovine source. We see here that insulin has two chains, two polypeptide chains. We have the A chain and the B chain, and we see the sequence of amino acids in each chain. In fact, in this representation, we also see the disulfide linkages. We see a disulfide linkage within the A chain, and we see two disulfide linkages between A chain and B chain. Now, the amino acid sequence is, yes, it's important because you know that the character of a protein relies more on its amino acid sequence than its amino acid composition. But as we go along, we see that this amino acid sequence, although it's of, we don't get much information from it, we see that the other levels, the higher levels of protein structure actually build on this amino acid sequence. Let's go to the secondary structure. In the secondary structure of proteins, we are now looking at the peptide backbone. So this is the chain, the part of the chain in your protein along which your peptide bonds run. Okay, so we are looking at the chain of peptide bonds. So we have the alpha carbon followed by the carbonyl carbon, and then the amine or the nitrogen in the amino group, which is part of the peptide bond. And then we have the alpha carbon again of the next amino acid residue. And then we have the carbonyl carbon, the nitrogen of the amino group, and then the next alpha carbon, and so on. That is what we are looking at. Now, if we talk about secondary structure of proteins, we look at what we call torsion angles or dihedral angles. So there are two important dihedral angles which can dictate the secondary structure of proteins. Now, a dihedral angle, in order to be able to detect a dihedral angle, you should count four atoms. So you should be looking at four atoms. In particular, you are looking at the alpha carbon of one amino acid residue, followed by the carbonyl carbon, and then the nitrogen of the amino group, followed by the alpha carbon of the next amino acid residue. So we have this. This one is defined as, if you are looking at the alpha carbon nitrogen bond, okay, if you are looking here, this angle is referred to as the phi angle. So let's say that you are looking at this direction. So you are looking through the alpha carbon nitrogen bond. Okay, now if you see that this part, the carbonyl carbon nitrogen, or basically this and this bonds, okay, these two bonds, if they are 180 degrees with respect to the alpha carbon nitrogen bond, then your phi angle is equal to 180 degrees. Now again, if you are looking through the alpha carbon nitrogen bond and you see that the carbonyl carbon alpha carbon, the next alpha carbon, is twisted with respect to the alpha carbon nitrogen bond. So it's twisted, let's say 90 degrees. Okay? So you see the difference. This time the plane, okay, if the carbonyl carbon and alpha carbon bond is twisted negative 90 degrees relative to the alpha carbon nitrogen bond, it means that this whole plane, the peptide bond plane, is 90 degrees, right? Or negative 90 degrees. So this creates another orientation of your polypeptide chain. Now, the next bond is called the Psi bond, Psi angle. Sorry. So the Psi angle is if you are looking through the alpha carbon carbonyl carbon bond. So it's the same way you are looking through this bond. So this is the alpha carbon, this is the carbonyl carbon, this is the bond. So you are looking through it, okay? Again, you are looking at the relative orientation of the amino nitrogen and the next alpha carbon relative to this bond. So you can imagine that you have two planes that can be twisted at the same vertex. So for me to imagine this, I usually get two sheets of paper. These are quite big, so you can use smaller sheets. And then imagine that these are the two planes, and I just twist them so I can imagine the orientation of the two peptide planes according to their phi angle and psi angle. Now, these two angles can be plotted on an xy coordinate plane. And in this plane, we can see that not all of the combinations lead to a specific protein structure. In fact, most of them are sort of not favorable structure-wise. So this is called the Ramachandran plot or Ramachandran diagram. And we will see here that the blue regions are those that are allowed. They are the allowed angles. The green ones are more crowded. They are possible, but they are not very stable. And the unmarked regions are the ones that are not possible at all. So it's because, why is there a limit? It's because of the steric clashes of your side chains, of the atoms in your, the atoms along your peptide plane. So this is actually quite hard to imagine, but if you can come up with a way on how to visualize it, like using two sheets of paper, or if you want, you can make some models out of matchsticks and clay, and then you can move them. You can do that so that you can visualize it. It's hard to imagine just in your mind. Now, the consequence of these dihedral angles lead to two common secondary structures. First, we have the alpha helix. So the alpha helix, if we look back in your Ramachandran plot, it's here. So this region, and we see that both of your phi and psi angles are negative. If you try to imagine this, so you have a negative 90-degree plane and then a negative 30 plane, another negative 90, another negative 30, and so on, you will notice that your peptide chain will assume a helical conformation. So we have this helix or alpha helix. So some observations with your alpha helix is that you need 3.6 residues to complete one turn of your alpha helix, and the depth of one turn is approximately 5.4 angstroms. Then if we look down an alpha helix, okay, we see that the side chains are oriented outward. Okay, so you see here the purple rings or the purple spheres, these are the side chains of your amino acids and they are outside the helix. Now in this model, this is called the ball and stick model. You see an empty space inside the helix. In reality, this is not true, okay? If we use a hard sphere or space filling model, we see that it is very compact in the middle of your alpha helical structure. And then finally, this is what we call a helical wheel projection of an alpha helix. In most alpha helices, it is noticed or observed that um, the residues that make up the beginning and end of one turn, most of the time, they are charged residues. In fact, they are oppositely charged. So, for example, if you have this sphere labeled 1, again, it is known that we need 3.6 residues to complete one turn. So, that is approximately 4 residues. So, if you look at the fourth residue, this time it's negatively charged. And why is this? This is favorable. Okay? So since they are on the same side of your helical structure, they have favorable electrostatic interactions. Sometimes you will also observe that on the same side of your helix, you will find nonpolar side chains. This is again favorable because your nonpolar side chains can interact via van der Waals interactions. And finally, I forgot to mention that your peptide bonds, the carbonyl carbon and the nitrogen hydrogen of your peptide bonds form hydrogen bonds with each other. So in this representation, you see the oxygen of a carbonyl group forming a hydrogen bond with the hydrogen of the NH of the residue that is 4 spaces away from this first amino acid residue. And this only tells us that an alpha helical structure is very stable. Now let's go to another secondary structure. We will go to the beta sheets. So here we see that in your Ramachandran plot, your beta sheets are due to positive phi and psi angles. So they are located here. Now there are two types of beta sheets. You have the antiparallel beta sheets and the parallel beta sheets. Now let's talk about the structure of the beta strand first. So the beta strand is basically a linear orientation of your peptide chain. And um, again, you can try to imagine the dihedral angles. And you will see that this will lead to a linear orientation of your side chains, the peptide backbone, and the side chains of the amino acids. Some of them are above the plane of the peptide backbone, and some of them are below the plane of the peptide backbone. In fact, they are alternating. Now, if you have several sequences that form beta strands, then you can form what we call an antiparallel beta sheet. So it means that the direction of one strand is opposite the direction of the next strand, which is opposite the direction of the next strand, and so on. This direction refers to the N-terminal, C-terminal directionality. Now, we can also form the parallel beta sheet. So in the parallel beta sheet, you have parallel directions of your beta strands. So again, these beta strands are found from different regions of a single polypeptide chain. So later on, you will see structures that form this type of secondary structure. Now, just to comment, your antiparallel beta sheet is more stable because the hydrogen bonds formed between your NH group and carbonyl group is more linear than the hydrogen bonds formed in the parallel beta sheet. Okay, so there is some angle in the hydrogen bond in the parallel beta sheet, but in the antiparallel, it's linear, so it's the optimum orientation for a hydrogen bond. Now let's go to the next level in the hierarchy. So we now go to tertiary protein structure. The tertiary protein structure refers to the overall structure of a single polypeptide chain. So it's a combination of your alpha helices, beta strands, and so on. Okay. Now, aside from the combination of these secondary structures, formation of disulfide linkages from cysteine residues far apart from each other in the sequence can also dictate the tertiary structure. Hydrogen bonds between residues far apart from each other in the sequence and are brought in proximity because of the folding of the protein that is also part of your tertiary structure. So these are the factors that dictate the overall structure of your protein. So tertiary structure is the three-dimensional overall structure of one polypeptide chain. So we see here several representations. These are combinations of your secondary structures. So here in letter A, this is cytochrome B from E. coli. This is composed of 106 residues. And we see that its secondary structures primarily alpha helix. In fact, we only see alpha helices that fold on each other, forming the tertiary structure of the protein. And within these alpha helices, you see heme, a heme group that is essential to the structure of this cytochrome B. Next, we have an immunoglobulin fragment. So this is just one part of your immunoglobulin molecule. In this one, we see beta strands, and in fact, there are some sheets forming. So we see here an anti-parallel beta sheet composed of this orange, um, sky blue, and green beta strands. So you see the directionality, they are anti-parallel. And then we have another beta sheet at the back. And if we look here, the one at the back is composed of the yellow, blue, and purple beta strands, and they are also forming anti-parallel beta sheet. Okay, so do you understand how to read this type of structure representations? Okay, so you observe that helices in these three-dimensional models, they are represented as helix, but for beta strands, they are represented as flat arrows. So the arrows are to tell us the direction of the strand. Now, in this two-dimensional representation, your alpha helix are represented by rods, and the beta strands again are represented by arrows. Now, here in another structure here, this is a part of lactate dehydrogenase from dogfish. It is composed of 163 residues. Now, for the three-dimensional model, you can easily see that we have a combination of alpha helices and beta strands. Now, for us to clearly see this, we can look at the two-dimensional representation. So we have the N-terminal here from the yellow one. Okay, so the N-terminal, we see a very short alpha helix followed by a beta strand. And then we have an alpha helix again followed by a beta strand. Another alpha helix, beta strand, beta strand. So we have two beta strands that are parallel. And then another alpha helix, another beta strand, another alpha helix, and another beta strand. Now, here are more complicated structures. So this one is a retinol binding protein from humans, and it is composed completely of beta strands. In fact, this orientation of your beta strands is very common in many proteins. It has earned its own name. So this is what we call the beta barrel structure. So it's because your beta strands form this barrel-like structure, and there is space in the middle of this barrel-like structure. So you see that your beta strands form antiparallel curved sheets. Next, we have here Asparagine Amides. So in this one, we see that there are beta strands interacting with each other. But if you look at the sequence, they are far apart from each other. So if you look here, from the N terminal, you have a beta strand interacting with a beta strand in the C terminal. So again, you see the structure of the protein folding in itself. Regions that are far apart in the sequence come into contact with each other. Uh, Again, we see another combination of the alpha helix and beta strand. So this one, it's completely alternating. Beta strand, alpha, beta strand, alpha, beta strand, alpha, and so on. And we see that the protein has this globular shape, and you have some space in the middle of the sphere of your protein. So this is your tertiary structure. The highest level in the hierarchy of protein structure is your quaternary structure. But before we go there, I would like to mention about domains. So this is a glyceraldehyde 3-phosphate dehydrogenase, an enzyme that is important in glycolysis. So if you notice in this three-dimensional model, you have two colors. It doesn't mean that you have two distinct polypeptide chains. This is one polypeptide chain. The two colors just tell us that there are two domains in this protein. Now, a domain is something that has its own unique tertiary structure. And most of the time, a domain in the in a single protein has its own function. Other domains in the same protein also performs a different function. So, for example, here, the N-terminal, which is colored blue, is the one that holds the NAD, which is essential to the function of the whole protein. So this blue domain is the NAD binding domain, while the substrate of the protein, which is glyceraldehyde 3 phosphate, is bound by this orange domain. So you see that there is sort of compartmentalization of the functions of the protein. Essentially, this enzyme has one function, which is to add a phosphate group to glyceraldehyde 3 phosphate. But it needs NAD in performing this function, and it needs its substrate. So you have compartmentalization in the protein. One domain binds the substrate, another domain binds the cofactor essential to the function of the protein. Now let's go to quaternary structure. When we say quaternary structure, we are talking about multi-subunit proteins. One subunit is one polypeptide chain. So, in proteins displaying quaternary structure, you have more than one polypeptide chain comprising its structure. A classical example is hemoglobin. Hemoglobin, this very big molecule, is made up of four polypeptide chains. Two of these are the same, and the other two are the same chains, and they form this structure. So you have the alpha chain or the alpha subunits. There are two of these. And you have the beta chain or the beta subunits. There are also two of these. Okay, so the yellow and the green, these are the two alpha chains. And the blue and the cyan, these are the two beta chains. So here are some terms associated with proteins displaying quaternary structure. So if we say it's a dimer, then it means it has two polypeptide chains, trimer, three polypeptide chains. So hemoglobin is a tetramer. Now there are bigger proteins composed of more than four polypeptide chains. There are some proteins composed of eight polypeptide chains. Okay? In that case, we can call them oligomer. Then we also have these two terms, heteromer and homomer. So if, for example, we have a dimer and the two chains are the same, they have the same amino acid sequence, then we call that dimer specifically a homodimer. Now, if the two chains are not the same, they have different amino acid sequences, we can call that heterodimer. So hetero, if you have different sequences for the chains comprising your multi-subunit protein. Homo, the prefix homo is used if you have same chains comprising the quaternary structure of your protein.